Okay.
Good afternoon, everyone. Welcome back to the second session of the workshop. Hope you all are enjoying it. For the next session, we have few guidelines for the participants. First of all, we request you all the participants to be ready with the desktop or laptops for hands-on session. Then we will take the attendance for each session, and the attendance sheet will be available during the lecture in the chat box as well as in the description section of the lecture. Minimum 80% of attendance is essential for receiving the e-certificate. Also, at the end of the workshop, there will be a feedback form which needs to be submitted for receiving the e-certificate. Please fill your names and email addresses carefully. And we request you to post the queries or doubts of the lecture in the chat box. We would try to address most of your queries after the respective lecture is over. Now, moving forward, I would like to request Dr. Yasha Hasija, coordinator of the workshop, to please introduce Dr. Amit Mandel for the next session. Ma'am, please.
The world of biology has undergone a stunning transformation in the past 2 decades. Data analytics has become essential for biologists and are, in fact, at the core of scientific discovery. Getting started can be a significant challenge for those uncomfortable with computational analytics, mathematics, or statistics. We intend to change that with this week-long hands-on workshop on bioinformatics. Good afternoon, everyone. I am pleased to introduce you all to the second speaker of the day, Dr. Amit Mandel. Dr. Amit Mandel is a research associate at the National Heart and Lung Institute, NHLI, at Imperial College, London. His postdoctoral research is focused on the identification of genetic aberrations in tumor types arising from exposure to environmental carcinogens. At present in NHLI, he is also part of a team interrogating genetic factors behind a lung cancer type with a long latency period arising from asbestos exposure. Prior to this, he was working at Cancer Research UK looking at skin tumors arising from UV exposure. Dr. Mandel has completed his PhD from CSIR IGIB, Delhi, where he was first introduced to the human genome and the dizzying diversity of genetic elements inhabiting in it. He then studied how a class of mobile genes could affect messenger RNA diversity and expression dynamics. I invite Dr. Mandel to take over the session on interpreting the transcriptome state from NGS studies. Dr. Amit, please.
Sir, you are unmute, so can you please?
Right. Thank you very much for the very kind introduction, and good morning to everyone. I'm Amit, and as you have heard already, my postdoctoral research is focused on understanding the genetic factors behind why tumors arise and why tumors go on to develop in some patients, and in other patients respond to treatment. So this workshop session is intended to be session on analyzing transcriptome data. But before I go into or we go into the details, what I would like to do is get some background made for the data sets that we are going to look into and what is the motivation behind it. So as with the gene expression data sets previously, or when it started with high throughput experiments, people were doing microarray or high throughput oligonucleotide arrays, and gradually, and in fact, in the recent years, sequencing has overtaken microarrays, and not just next generation sequencing. Nowadays, you have single molecule sequencing or ultra long read sequencing of RNA. But for this workshop session, the idea is to look at the most widely available next generation sequencing data set or platform that is being used at the moment, and that is the Illumina platform um, uh, for RNA sequencing. So having said that, so we are going to look into analyzing data sets arising from RNA sequencing from the Illumina platform. But because this workshop, we are not person and not meeting in person, and this is happening online, and so the way I have designed or tried to arrange this workshop is most of the hands-on, or in fact all of the hands-on, would be happening on the R language platform using the R Studio software environment. So there are some limitations which I should go into first. So as with high throughput data sets, especially next generation sequencing data sets, the size of the data sets are quite large, and they are not amenable or feasible so that you can look into it in those files in a text editor, or you could analyze them on your laptop or your standard desktop PCs. Rather than that, what is required, or what happens is that you need high performance computing environment where you analyze those files. And once first stage of analysis has gone into that, which is the bigger part of the data churning process, whereas server side environment is required, the second stage data that comes from the next generation sequencing experiments is in case of RNA sequencing, are the count values. So once the raw data has been aligned to the genome or the transcriptome, what we get is count values for the genes, and that's the stage from where we are going to start from. We are going to start from the count values for the genes, and because these values or these files are can be analyzed in an R or R Studio environment using your laptop or desktop environment. What I had a couple of other points written down. Right. So hopefully you would have seen there is a link that has been posted of a web page where I have posted the data sets that we are going to use for this session. The data sets that we are going to use for this session are from the TCGA what you can say consortium. The TCGA consortium has been a very large multinational collaborative effort on cancer genomics, and it has analyzed more than 30-40 different cancer types in hundreds or more than or even in some cases around thousands of samples, and the data sets that have arisen from these for this collaborative effort range from mutation, gene expression, copy number, methylation, protein arrays, and the like. And these data sets are available for in the public domain, and the website from where these data sets can be made available or you can access is the NCI GDC portal. In a short while, I'll give a brief walk around the NCI GDC website and how to access the data set from there. And then we would jump into the R Studio session. Because as I understand, there many of you might be already using R Studio, might be already using packages in there to analyze different data sets, but some of you might be quite new to R Studio or maybe not have used R Studio before or the R environment before. So before we start the analysis steps, I'll give a short uh, introduction to the R environment and the R Studio, and also you would have seen in the schedule for the workshop. I think on the second or the third day, you have another session using R. So that they should come helpful on that day. Okay, so so I'm going to share my screen now. Hold on a second. Is my screen sharing visible to everyone?
No, sir, not yet.
Okay. Is it now?
Yes, sir. Now is it
Okay.
visible?
Thank you. Okay. So as I said, so if you Google NCI GDC you would reach on this page, which is which hosts the data sets from cancer genomics, um, and here um, if we go to the repository, I would give example of how to go about downloading one type of data set and any other variation, any other different cancer type would be similar. So if we go to the cases tab, and there are multiple windows to filter the data. So the first we can, if you know the name of the project, you can directly scroll down to the project tab here and select which is, so in our case we are going to be looking at the adenocarcinoma cohort, lung adenocarcinoma cohort, which is whose name, short name is TCGA LUAD. So there. So TCG LUAD, either you could select that, or if you're not aware of the name, you can expand the primary site, and you can select, let's suppose in this case, I'm looking for lung related data sets. There is a text box search. Okay, I can't seem to spot the lung tissue. Anyways, since I'm aware of the project name, which is TCGA LUAD, so if we select the project here, so once the project is selected, so you can see on top that it shows that the project, it shows the filter that has been applied. And then once that has been applied, go back to the Files tab. Then the data category, if you expand it, you can see that there is transcriptome profiling. So if you select transcriptome profiling and then select gene expression quantification, and then you have the count values, HT-sec counts. There are two other types of two other variants of count data, but we would not be using those because the statistical methods that we are going to apply would be amenable with count data. So once you have selected counts, so now you can see that there is another the bottommost selection window says open, which means the files which are under open access. That means you can download them directly. All of them are under open access in the count data. There are files which are under control access here, which are sequence, direct raw sequence data or BAM files, which of course we cannot you cannot download unless you have applied for a access key. So once you have done that, we can select add all files to the cart. Once you have done that, then you can go to cart on the top here, and that shows you the number of cases that are present, number of files, and the total size. So you could download these count files from here, and also the clinical data, related clinical data from here. So this particular data has been downloaded and has been formatted for this workshop, and that is present on the website that I have, the web page link that I have given. So these are these were the two data sets. So in the case of this particular project on lung adenocarcinoma, there are around 500 tumor samples for which we have expression values per gene, and then we have around 50 odd control tissue samples to compare and look at differences between tumor and control. So this is the data set that we are going to work on today. These are the link that you could download, and now I would start with the our session. Before I go into the R studio and analysis steps, there is one more thing to mention, that though the R language or the language or the analysis platform that we're going to use, R Studio is can be installed on whether you are on a Apple Mac operating system, or on Linux operating system, or a Windows operating system. And I understand that many of you would be on different operating systems using laptops or PCs. But many of the packages within R that we are going to use would not be available on a Windows operating system. So though you can have the base R installation or R Studio installation, you might not be able to. You would not be able to install that package. So in that case, I would strongly recommend that in that in many of the research institutes and universities, we have the R Studio environment hosted on your institutional computation server, where you can access the R Studio as within your web page. And because that would be hosted on a Linux server, when you would be using that, you would be able to install packages that are most of the packages that are used for the kind of analysis that we are going to look at today. If you are already on a Mac or a Linux operating system, then then it is then it's all right. You could, if you already have R Studio installed, then great. We can start right on. If you do not have R Studio installed, I would strongly recommend that please install it, so that together we can go step by step across the commands. Um, Along with the data sets, I have also on the website page, you would see that I have also given command history, or the actual commands that have been used um, um, for, or that would be used for this session. Um, and uh, and I have annotated. I've also put comments or annotation on those above those commands, so that even if you are not completely at the same stage where the analysis is going on, you can still hopefully catch up. And and apart from that, as you would see, what I have done is at each stage there are breaks in the analysis stage. So at each stage, the files that are required, I have also uploaded that on the website, which you could join the analysis step midway, rather than starting to follow through from the beginning. So so what I have tried is given help in the command files, and also given lateral entry points, and which I will describe them as I go along. Um, okay. So having said that, the limitations about that we are not we are not doing hands-on on a server. We are going to look at data sets using RStudio, and on top of that, the packages that we are going to use might not. In fact, two of the main packages that we are going to look at is not available on Windows based operating systems. Um, um, and after that, there is one more thing that I wanted to introduce, and that was the Anaconda, or Conda environment. Um, if you have already heard about Conda environment or the Anaconda repository, then that's great. You're already onto it. Those of you who haven't heard about Anaconda or the Conda repository. It's a Python distribution, Python language distribution, but it has become something much bigger than that. And by that I mean that if you uh, install the Conda environment on your laptop or on your desktop, then what it allows you is that it allows you to install a large number of bioinformatic related softwares and packages, not necessarily on Python language, but it could be any other language. But many of them are being hosted on the Anaconda or the Conda environment. And what happens what it allows you to do is that when if you are, let's suppose, on a Mac or a Linux operating system, and you want to install a particular package for analysis of or maybe alignment or maybe plotting or some other analytical purpose, and if you the moment you want to install it, there would be some dependencies, and you would have to worry about uh, to install all the dependencies. But having the Anaconda environment, what it allows you is that you could simply like on a like you can search for the package. So if I so this is the Anaconda package repository. So let's suppose I wanted to install BWA, which is an alignment program. Um, So that's the command that you need to run on if you have the Conda environment installed on your system. And what would happen then is that the Conda environment would get all of the dependent packages and installation or whatever that is required to get BWA installed on your system. And here you can see that this is available on either Linux or the Mac operating system. But um, if you are on either of those system or on a server side environment, it makes life a lot more easier. So okay, so that was the introduction about Conda environment, and I said that because that's the environment that I'm using. So this is I have the Conda environment installed on my laptop, and on that I have RStudio. Um, so service. I'll just increase the font size a more. Um, Hopefully that's better. Okay. So this is the R environment or the RStudio environment. If the R environment can also be accessed on command line, and R also has its own console, graphical console. What RStudio is, it's you can say R with added steroids or with added jet boosters, you can say, because it allows you, or it helps you to make an environment where you can create or perform uh, analytical tasks that can be documented thoroughly, comprehensively, and because you can document them, and then it also allows you to share them for reproducibility, which is a lot of which is quite important in analysis. Okay, um, so So what I have done just now is that I have gone on File and from the menu, File, Open File, and then I have pointed it to the first command history that is available on the website, on the web page. So these were the starting data sets, and here you had the R code. So if you download that R code from this link, and then open it from File, Open File, and point it to the path where you have downloaded the R code, and then it opens like this. And what then you could do is that you can simply, if copy the command and or select the command, and you can simply click here, run, or you can copy and paste it here on the main console, and then hit enter, run. So this is the advantage of our studio environment. You have multiple windows which each with a different purpose that allows you to get your work done. So this is the window where you have the command. If you have a command history or a R script file that you could open, this is your main console where you can type. This particular environment shows the data sets or the variables that you have created. And here, this particular environment, the particular window, you can look for help. So if you click the help tab, you have different whatever packages are installed or available within your R Studio environment, you can search for help here. And then the packages tab shows the packages that are installed within the R Studio environment. Okay. So we are going to start. So the command that I have uploaded, it assumes that the two files that you have. So if we go to so these two files, tumor counts and the count values from the control tissue, they are in the same directory where when you have opened your R Studio, that the directory where R Studio has opened. To know which directory R Studio is or what is your working directory default, the command is get working directory. So getwd and then an empty bracket, and that tells you that's the working directory. If you wanted to set the working directory, so that is set wd, as in set working directory. So I'm going to go to the now if I do get working directory again, now it should show me that that working directory has changed. Whatever command you type once on the R Studio environment, you need not type it again. If it's the same command or an iteration of the command, you can use your up or down arrows, and you can circulate between the commands that you have typed already in the current session. Okay, so let's start. So we are going to read the count data for the tumor and the control tissue. So as I said, you can, if you have opened the command file within the R Studio session, and you have selected this, you can do like this. You can say run. Once you click run, that has so it's running, and it's a large data set, so it takes a while. And when you have enter the command and the studio or the R is processing, you can see this red button over here, which indicates that, okay, so I had clicked twice, so that's why um, the, the command had been entered twice here. But that's the idea. You can select here and say run, or you could copy. You can select. You can copy it from here and you can paste here. Command C, Command V, or um, um, if that's easier for you, you could do it like that as well. So I've done the another command. Okay, so now we have read both the tumor and the control tissue um, within the R Studio environment, and so the command that we have used is read.table, and the advantage of using R Studio environment is that whatever function you are going to use, you can, if you are not aware of what that function is, has different parameters, and what those parameters are doing, and or what could be done using that function, you copy that function name and put in this search box over here in the under the help tab, and 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 you can have it opens up the help page for the read.table, where you have got the detailed description of how to use that function, what are the different parameters present here, and also if you scroll down, there would be couple of examples as well to help you get going. So that's the advantage of using RStudio, that it's quite helpful or intuitive in learning if you are new to this R or RStudio. Okay, of course, because this session it is we are not solely dealing with R or RStudio. We would not be able to go into detail with of each and every command and its syntax and what it means, and how it is, what are the meaning of the different parameters in RStudio. But having said that, whenever there is a particular step which has a or an important step, I'll go over that in detail as to why we are doing what we are doing, and what could be the other options to it. So at this particular stage, I have just read the data. I'm trying to go much slower at the moment, but gradually as we move along, I'll try to increase our pace, so that we are able to gain as much as we can in the time that we have. Right. So so what we have done is we have read the tumor and the control tissue data sets. And as I said, this is from the lung adenocarcinoma tumor, from the TCG collaborative project. The particular cancer type that we are using is not relevant for this workshop. I had to use a particular example, and I chose this example because this was which I could found, which had a large number of samples, tumor samples, and also additionally a sizable number of control tissue samples, so that at the end, when we have learned a bit of how to import the data in R and how to normalize it, then we can do relatively straightforward differential expression comparing the tumor with the germline or the control tissue. When I say control tissue, these control tissue, I mean this is tissue, normal tissue adjacent to the tumor from the patients. So that is good. So when you have any data within R, you can peep into it, as in you can look into it, what is present in it, and one of the basic ways to do that is you can say dim as in dimension, and if I say dim dd tumor, you can see that if you have written a couple of letters, RStudio prompts you as for what variable you wanted to type. So you can see that it is showing that did you want to type dd tumor or dd germ. So that's there. So let's suppose if I do this, so it says 60483, and then there's a space, and it says 5502. So in our the convention is that the first number that is shown in case of dimensions are the number of rows, and the second number is the number of columns. So what that means is that this is a two-dimensional data set, or a data set which has around 60,000 rows and 502 samples. These rows, each of these rows correspond to a unique gene. We can look at the if I another way to look into it is if I can say write the object name and put a square bracket. Square brackets in R is a way of subsetting. So by subsetting, I mean if you have a data set which has multiple number of columns and multiple number of rows, and if you wanted to look at a particular number of rows from particular number of columns specified, then you can write that here. As I said, the first number is for rows, and the second number is for columns. So if in the syntax for subsetting, when you put a square bracket and there is a comma in between, before the comma it is rows, and what 1:3 or 1:3 means, I want to look at first, second, and third rows, and after the comma it's 1:3 again, which means I want to look at the first three columns. So that particular subset of the data, so first three rows and first three columns. So this is this is what we are looking at the data. And similarly, the data is in the same structure in case of the germline. And these values that we are looking at, these are the count values. And this is the particular gene. The gene ID that we are looking at, so this gene ID is an ensemble gene ID. So that is the ensemble database. So there are a couple of databases that we should be aware of if we are analyzing genomic data sets, whether even if it's not human, it's a mammalian, or even if it's other organisms, non-vertebrate organisms. So that's an ensemble database. That's the other is NCBI, and the third is UCSC. These three are quite big in terms of their coverage of the organisms and the data sets that they host. Ensemble is quite preferable, I would say, in case if you are interested in, or if you are working on human or mammalian organisms, because especially in case of humans, if you are working on data sets that where you are looking at variants or mutations, the ensemble annotation resource is considered to be a standard resource. So in that context, working with ensemble related data sets, annotation data is sensible, makes sense. So we are looking at ensemble gene IDs. So each of these IDs correspond to a particular gene, and these are the columns. So each column corresponds to a particular sample, and on top of it, we can see that's the sample sample ID or the patient ID from the TCGA database. Similarly, it would be, so if we look at So now we are looking at the similar, the structure in the germline case. So that's, and again, these are the counts. When I say counts, so how these counts are obtained. So that is what I discussed in the beginning. Because we are going to work, we are working on our studio, not starting from the raw data. So when we start from the raw data, when we get the sequence, those are present as fast Q reads, which are aligned using specific alignment programs. And when those FASTQ reads are aligned to the genome, the algorithm, what it does is it aligns, it reads at genomic position, and then it also looks at the annotation that you have given. So there are two different kind of annotation that goes into when you are aligning um, RNA sequencing data set. One is the information of the genomic sequence. So you give a FASTA file of the genome, and the second, which is quite important, is the transcriptome annotation. So the known annotation, the 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 coordinates of the genes that are known in that particular organisms. And most of the organisms which are which are being worked on in the research community, they if they have the genome sequenced, like the mammalian or many of the other vertebrate, or even like C. elegans, you would have reference gene annotation available. And that gene annotation is available in the form of a form called as GTF. So that's a gene transfer format, and that annotation goes into these alignment programs for RNA sequencing. So the alignment program looks at the genomic sequence and the gene coordinates from the GTF file, and then it tries to make out that if in this particular gene there are 100 reads aligning, then that, so the count value for that gene would become 100. So that's in a very simple language. It probably means that for this particular gene, there had been around 1535 reads that had aligned, whereas for this particular gene, maybe 2 reads had aligned. So that is the the idea behind looking at count data sets. Okay, now let's move ahead. So what we are going to do next is, because so you can see that the gene IDs have version information, version information. So you have the gene ID and then decimal 13, which means that this is the 13th version of this particular gene. And so these annotation databases keep getting regularly updated, and when they get updated, they can have version information in those IDs. At this particular point, or what we are going to do, looking at gene expression, normalization, and differential expression, we do not need those version information. So we could remove that. And this particular command here is stripping those decimal points out. And I'll briefly go over what it does. Is this is the command. This is gsub, as in it's a substitution command. What it does is, we are saying that within this double inverted commas, I have given a pattern, regular expression pattern, and that pattern, when it finds, it would replace it with within this, whatever is within this double inverted comma. And you can see within this double inverted comma over here, there is nothing, which means that whenever I find this pattern, I want to replace it with nothing. So, and the pattern that I'm looking for is that I'm looking for whenever there is a decimal point, after that, any number from 0 to 9, either present once or twice. So it may be 5, or even if it's 13 or 20, it would be replaced by null. And since I have done that, I can I'll do the same command as I've done before, and I can see that it has now stripped away. So did the same um, um, for the control tissue samples. Now, what I'm going to do is that I'm going to combine the tumor and the control sample data set together. And to combine them, I'm going to use this particular function merge. You can see, look at the help page for merge function. What it does is that the idea behind using merge is that, so let's run this. Um, the idea behind using merge is that you can give two data sets, and if you want to merge them, there should be a column or a key, or you can say in another language, you can say a particular column which is the same in both the, or has the same IDs in both the in both the data sets. So I'm saying merge these two data sets, and by x equal to 0, 0 is for the row names. So this first column is actually row names. These are the column names and these are the row names. What it means is that here I'm saying that merge these two data sets by the row names for each. So it would find out for this particular gene, it would see what are the values in this data set and what are these values in this data set, and it would put the columns one after the other combined. So, so 502 samples and 59 samples combine, and it becomes 561 samples. But we can see that it's become 562, and the reason is that when you merge, the row name is lost. The row name instead comes within the data set. So so we would take, so you can see if I do it like this, I can peep into the column names that I have created the merge data set. I can see that this, or I can also look into that data set like in 1:3, 1:3. And I can see that the row names have gone. Instead, the row names have now become a column. So I'll take the row names back to where they were. So that's this step is doing. I hope everybody is, if you are, I mean, at least up to this stage, we have not yet started using any package which is not available within Windows OS. We are using the base version of R, and this should be working on Mac, Linux, Windows, any of them. So now we have created the data set. There. So now we have the data set where we have 561 columns, so 561 samples, and now we can do some, we can delete the data so that our slate is clean. So now we have the data to start with. Okay, now comes the, from here, these two packages are not available on Windows OS. So if you are using a laptop with Windows or a desktop with Windows OS, I'd recommend that if you are able to access a Linux or a Mac OS or a server operating system or a HPC and operating HPC environment, I would definitely recommend using these command history on there and try your hand on these commands. So we are using these two packages. So the first package, EdgeR, we are going to use this package to estimate the library size and remove genes which are very under low expression, and then we are going to use this particular package Lima, which is for differential expression. So Okay. So what so the- so so a lookout or a heads up of where we are heading to. So we have now a data set of 561 samples of count values for each of the genes. So there are 60,000 genes across 561 samples. And before we try to draw any kind of conclusion from this data set, what we want to do is that we want to first normalize the data set. Why do we want to normalize it? We want to normalize it because these sequencing, these are separate samples, and these samples have been sequenced in different batches, in different at different time points, and also not all of these samples would be of equivalent library size. What I mean is that there might be samples which have low sequencing depth, and there might be samples which are okay, and there might be samples which are very high, and we need to know that before, so that we can normalize comprehensively. So that's what this command is doing. So that creates a digital gene expression class object, and once this object is created, we can get an idea of the library size that is. So what is happening here? So once you create a DGE list object from the jar package, and you do a summary of the library sizes, you can get an idea of the distribution of the library size across the samples. We can see that the median library size is around 46 million. This is 46 million. So we can also look at what is the distribution of the library size. Like, so here in this command, whenever there is a plot being made, in the command history, I've tried to save it within a PDF so that you can come back to it later on and look at it. But here I can just, okay, I've missed a command. So I've got an error message here. I've got an error message because a particular function is a particular command step is missing, and that what is missing is this particular object is missing. I'll create this object. So we can see that there is a lot of variation in the library size that we are looking here. Um, um, so, so ideally what we would want to do is that we would want to consider that information when we are trying to normalize the data. So So let's try to find out how many samples are at the extreme ends of the library size. So this particular command, what is this command? What this command is doing is that we have a particular object. So this object is we can check the dimensions of this object. So it says it has 561 rows. So each of the row is for the sample, and then it has columns, 3 columns, and so if you can do head. Head allows you to so these are the columns that are present, and this is the library size column. And so this particular command, what it is doing is it is asking that for this particular data set in the column library size, how many samples are present which are less than 25 million. And when you do that, you get the value is 20, which means is that there are 20 samples which are less than 25 million library size. Similarly, if there are, if I there are 26 samples which are greater than 90 million library size. The idea behind doing this is that we need to find what are the samples which are at extreme ends of the library size. Okay, so this particular information has been this particular information. Um, this particular information has been incorporated in a sample sheet, which is which is present where I have provided the library size class, as in anything which is greater than 90 million library size is classed as very high. Anything which is less than 25 million is classed as very low, and anything and all that is in between is in the range. So So what we have done is we have read the sample info within the R environment for data sets which are small in size. So in this case, we have 561 rows and only 8 columns. It is convenient to, if you click this particular data here, there is a window that opens that can be, you can scroll as if you are in an Excel sheet. So if I click it here, so that shows me the data, that the way that's organized. You could try clicking this one, but because it has so many rows and columns, it would probably freeze. It might. So this is the sample sheet where you have the sample IDs, so 561 samples, and then the library sizes that are present, and the sample class, and then the library size class. As I said, anything which is large than 90 million is or smaller. So that is present. Apart from that, there are these batch information that is provided in this sample sheet, and this batch information has been taken from the TCGA batch effect viewer website. What that batch effect viewer website, it has that information that which of the samples had been from which tissue source center, as in tissue source center, because these projects have large number of samples which have been collected from multiple hospitals, and then they have been sequenced in different batches. So that kind of those information are available from also from TCG, and those these information can be used to try to correct any batch effect and perform normalization. So um, this is the command that is given here. So that particular command table, what it does is that it tabulates any character value if it's present in a column into the groups, and then it would say how many values are there. As we've seen earlier that there were 20 samples which were low library size depth and 26 which were higher, and then the rest were in the range. So that is being said over here. This basically is cleaning the environment, because now we know the library size. We've looked at the library size already. So let's clean that. Okay. This particular sample info was missing. At, at, I'll attach it on the class attachments. So I've added the sample sheet, and you could read that in. Okay, so we now what we have is that we have the combined data set and we have the sample sheet. Now we want to proceed with normalization, but there is another step that is required in normalization, and that is we would want to remove genes which are expressed at low levels. And why would we want to do that? And we want to do that because RNA-seq has a very high amount of values which are at the zero level. So let's suppose if I look at this, so this is the data or combined data set, right? And if let's suppose I make a density plot. Um, So I made a density plot for the first sample, and you can see. So on the x-axis are the range of the values. So what I have done is that I have log transformed the values, and you can see that there is it's kind of bimodal distribution. There is a huge peak which is at zero, around zero, and then there are values, of course, which go on range. But there is a peak which is at zero, and this is something which happens with RNA sequencing data sets, that there are many values which are at zero because this is sequencing related, and then what the alignment program is doing is that it has the annotation file that you have given, which has all the genes that have been detected across over the years in different experiments, and not necessarily all the genes that are known in the reference annotation. Let's suppose 50,000, 60,000 genes, not protein coding genes. So like protein coding genes plus non-coding small RNAs, pseudogenes, and different other types of genes. So not every gene is going to be expressed in a particular sample or a tissue type, but when the alignment program or the count data is being generated, you would get a value for all the genes that are present in the annotation file. So if your annotation file has, let's suppose, 60,000 genes, you would get a value. And even if there are no alignment or data present, you might get a zero in there. So there is a large amount of values that are zero. We want to remove genes which are mostly at basal level expression, so that our data set becomes much stronger as we go ahead. And to do that, to remove the to find out genes which are under low expression, what we are going to convert the count data into CPM format, which is a count per million. So basically, if let's suppose you're looking at this particular section here. So the first gene over here has 8,000. So the count value is around 8,500. So what the idea is that you sum the values for all the genes in this particular sample, and then express this 8,500 in terms of million. If the count was 1 million for this sample, what would have been the count value for this? So that is the basic idea of converting the values into CPM. It allows for manipulation, which I would just show. So what this has done is that now it has converted all the values to count per million. Now what I can do is that I can get an estimate. So the estimate is, let's suppose I want to filter out low expression genes. So, but I would need to have a threshold. So let's suppose my threshold is that I need at least 5 counts for a given gene. So, and we had seen earlier that our median library size was 46 million. So how much would be the count value needed, CPM value needed if I needed 5 counts? So that would be around 0.11. So that would be my CPM threshold. So that's the command over here. I'm saying that my CPM threshold should be greater than 0.11, by which I mean that genes should have at least 5 counts present. And for how many samples? So I have written over here 28. 28 is just 10 is around 5% of the total cohort size. Again, these are arbitrarily set thresholds depending on the data set that you have, and how good or bad is the data. Depending on quality, you might want to tinker with these thresholds. So what I have done is that I have said that I want at least 5 counts for a given 5 count for a given gene in at least 5% of the samples. And after I've said that, if I do this summary, what it tells me is that out of the 60,000 odd genes, around, in fact, more than 50% of those genes are get excluded because they are not expressed at sufficiently considerable level, not high, just considerable even. So not present at 5 counts in more than 5% of samples. So now what we would do is that we would subset our data so that we can remove we can select out only those genes which pass our criteria. So what I have done here is that we had this particular data set which had all of the gene and the total samples, and I have put a square bracket and initially said that a square bracket is for subsetting, and before the comma I have put this object, the keep object, which has the logical values of true and false. So what is happening is that for all the rows, as in for all the genes which has got a true value, it gets extracted out in this particular in this particular object. So that's the so that ways we have been able to subset our data. So now we can see that this particular, if I do dimensions, so it has now 31,000. Yeah, so 31,000 genes which have been selected because they passed the threshold. And after that, these two steps are moving ahead with the normalization process. Once we have the final set of genes that we want to carry forward, we create the DG list object again, and then we are this particular command or step. What it is doing is calculating the size factors for the library. Okay. So now we are ready to apply the normalization step. So when we want to, so we are going to normalize using the Lima package, and within Lima package, the normalization happens through a model matrix command. So what that model matrix command is meant to do is that it is meant to provide a formula which tells that what are the different variables in the data that need to be adjusted, or that and in what order. So this is the model matrix command. So here what I'm doing is that first adjust for ship date. So these are the batch effect variables. So what I'm saying is that first adjust for batch effect variables 1, 2, 3. So After adjusting for batch effect variable, shipping date, and tissue source center, then look at adjust for the library sizes, and finally then look at the sample classes, which is the tumor and the normal. So this way, what I mean to say is that after adjustment has been done for the batch effect variables, then look at the differences between the tumor and the normal. So that this is what the formula is intending to do. After we have built the formula, then we are going to run it. Again, the command that is being given there is it's saving it in a PDF, but you could of course run it, and then the graph would be plotted in the console. Right. So this is so these each of these small tiny dots represent one gene, and this is the red line is the line of fit, and what it is signifying is that more or less the algorithm is able to fit its variance adjustment function on the data distribution. If we had not removed the low expression genes, you would have seen a sharp beak here on the zero towards the zero at the x-axis towards the zero at the x-axis. There was a beak which should have been coming down, and the red line would not be able to fit. You could try the normalization step rather than using the 31,000 genes. If you use the total subset, total genes, you would see that effect. And I would encourage you if you wanted to test it out. Okay. Once we have done with the normalization, so that this is the particular step, the normalization has been done using this boom function. So where we gave this was my gene expression DG list object. This was my formula. That this formula was telling what things to adjust for and in what order, and then plot equal to true basically means that I want to store or visualize the plot, the boom plot. After that, you can store the normalized data out. You can extract the normalized data out. In case of objects in R, if there are, if it's a class list, then dollar is a way of subsetting. So if I put dollar, then all the lists that are that are present within that object, if I say dollar, so it is telling me that within there is a data called targets. There is E. If we look at the help page for boom function, we can see that the normalized expression data is within the E object. So that's what we are extracting out, and that is it. We have extracted out, and we can also look at that. So these are the values. And if I look at now, how do the values look? Now we can see that the values have changed significantly. Initially, they were in whole numbers, but now they are in decimal points and in across the scale. If I do the plot density function again, so now we can see that initially when we were seeing was there was a sharp peak at around zero, and so the data was so the whereas now what has happened is the data is kind of distributed around zero. So there is the peak around zero is no more there. Rather, you have again bimodal distribution but the two humps are kind of straddling zero in between. So that was the idea of removing the genes which had in majority very low expression. Finally, there is a command there to store or save box plots before and after. You could say that, and we can also write out the save the once we have the normalized data, we can write that out and save it. So write.table is the function to save any object that we have within the R environment. And so you give the object name and then within inverted commas the file name, or if it is in a different directory you want to save the path also before the file name, and then a couple of other parameters as well. I'm not going to save it. Finally, cleaning the environment. So RM is the function for removing. So RM is short for remove. If you have been working, or if you have worked on the command line environment in Linux, many of these commands share similarity in their name or syntax, at the least in their function with the commands that are available on the Linux or the Mac operating system. So RM is with RM function you can delete the objects from the environment. That's the deleted. The final command is save command, by which what you could do is you could save specific objects from the R environment within .r data file. And why is this useful? And the reason I have done this is that let's suppose when we move on to the next stage, even if you have got stuck somewhere in the middle of these commands, but you want to follow the commands of the next stage 2 commands, or you want to get to the results, you can just load this particular file in your environment, and you would get these objects within the R environment, and you can inspect them as to how the result files look. So that's the end of the stage 1, where we have normalized the data. Okay, so what I'm going to do is I'm going to close this RStudio session, and I'm going to open a new session with the stage to start the processing or for the stage 2 results. Going back on, so I will add the stage 2 files. Okay. So I've added the R code for the next stage. In the next stage, what we are going to do is we are going to look at the effect of batches that are present. We have already normalized considering the batches, but we are going to inspect it. So that if you come across a data set where there is a substantial batch effect, how to first visually inspect if there is a batch effect that is going on, and the method that is available or that could be used to remove any sort of batch effect that is present. So that is what we are going to look at in the next stage. So I'm going to close this session, our studio session. So as I said again, I'll reiterate my point again. So on the web page, you have the starting data set on the first page, which says start here, where you have the tumor counts and the control tissue counts. When you download them, they are in zip format. Unzip it, and then go to stage one and download the R command, R code, open it with an R Studio, and you could follow through the commands. There is one missing. Uh, um, Probably the sample sheet should have been, the sample sheet was not present, so the sample sheet I've added. So, and once you have gone through the stage one where we have normalized it, the final outcome is given as this R data file. So even if you want to start from this stage after the normalization, you don't need to download this particular R data file and load it into R Studio, and then you're good to go for batch effect correction. Similarly, so this is the stage two where we are going to do batch effect. So this is the R code that we are going to use. If you could download, and once we are done through with these with this R code, the outcome or the files that would be available after this session is present here.
Thank you, sir. That was a really nice presentation.
I'll just start with the next stage in a couple of minutes.
Yes, sir. Sure, sir.
Code file. And what I'm doing is now I'm loading the results of the stage one outcome, which is the R data file that I've given. And once I loaded, so I can see that the results are now in my working environment. So that's the advantage of storing objects as .r data. You can um, with a simple command of load, the objects get loaded into the environment. You don't have to read them one by one using read.table or similar commands to get them in your working environment. You could use the load command. It's also this R data functionality. R data functionality is very helpful if you are uh, working on on an analysis aspect, and you have created you are halfway through and you have to stop it, or come back to it later on. This is another good way of course, there is another way where you can save the whole workspace. But if you wanted to save specific objects which you would want to later on work on again further, so save them as .r data file, and then you could just load them into your session and then start working on them where you left off. Okay, so in this stage, what we are going to do is that we're going to look at whether there are any batch effects present in the data. So what we are going to do is first we are going to look at the raw counts where we had done no normalization, and then we would look at the womb normalized counts to see if the batch effect did decrease a bit there, and then at the end we would look at the package, SVA package. So all of these packages, as I said, I'm using most of these packages are part of the bioconductor environment. Um, those of you who are aware of bioconductor, that's quite good. If you are not, this is a very useful repository of packages specifically designed and maintained with very good documentation and regular workshops or conferences around bioinformatic related strategies. Um, I'm loading these three packages. So what I'm going to do is that I'm going to use principal component analysis, PCA plot, to look at how the samples are affected by batch. And once I have done PCA, what I would want to do is that I would want to visualize I would want to visualize the plots as a 3D scatter plot, and the scatter plot 3D is that package that we want. And because there could be n number of batches that are present, to assign colors on the fly, like if you want to have, like if you want to color, if you want 5 colors, there are 5 batches and you want 5 colors, or maybe n number of batches and colors. So Viridis is a package which is quite handy to assign as many number of colors that you want, with minimum fuss. And finally, scales is a package. What it does is that it allows you to get transparency in colors. So if you are, so imagine a scatter plot with lots of points overlaying each other, and if they're colored, then what would happen is that they would probably look like a smudge. Whereas if you put in transparency and there, it looks much decent and appealing. So that's why we have those three packages. So what we are going to do is, so you can see that I have the normalized count data in my environment. From there, and also the raw count data. So first I would want to extract out the tumor samples for to inspect the batch. I'm not going to look at the batch in all the 560 samples together. that is tumor and normal samples together, because um, you would suppose that there would be anyway a strong difference between the tumor and the control samples. And even if there is a batch, maybe the difference between the tumor and the control sample might override the batch effect, and you might think that okay, the tumor and control are separating, so probably there is no batch. So that is why it might be a good idea to look at the batch, try to visualize the batch in just the tumor samples. So we are looking at just the tumor samples in the raw data. So what I have done is that I've extracted out the 502 tumor samples only. And here what I'm doing is that because these were count data where you can see there are zero values. What I want is them, I want to convert them to log scale. But of course, if you have a zero value and you want to convert into log, you would get an error message, because that would probably, that is infinity if you want to do log to 0. So that is the reason that this particular command, where what I'm doing is that I'm adding 1 to each value, all of the values, and then converting to log, so that so what is 0 again would become 0, but count values which are not 0 would get converted to log after addition of 1. Okay, so now we are going to, so what we are doing is we are applying PR comp. So PR comp is calculating the principal components. The PR comp is a function. You can look at the help page for a lot of samples. So it's computing. You can see the red button over here. It shows that R is busy. Right. So here is the PR comp help page. I would recommend to go through it. So we have calculated we have done the PCA using PR comp function. Now what I want is what I'm doing is basically I'm taking out the plate ID. So I'm using plate ID as the main batch effect. So what plate ID means, as I said before, is so there are when samples have been sequenced in the TCGA for a particular cancer type. So lots of different hospitals have been involved, but there have been a couple of, or maybe one or two places where the sequencing have happened, and the sequencing happened when and that which samples have gone to get sequenced in what order, you could get that from plate ID, which is available from the biospecimen data type that you could download from NCI portal, or as well as from the TCGA batch effects viewer website. So that plate ID means what was the what was the order of the samples that went onto the sequencing machine. So that I'm extracting that information out, and using that information I would color the samples, so that if there are samples of a particular plate ID which are behaving differently rather than biology, but rather than biology there is the samples are clustering together by plate ID, you would be able to see that effect. So I've extracted the plate ID, and then what I have done, I'm converting it into a factor. Once you have converted a character class into a factor, you can then assign colors to them based upon their levels. So here I'm checking how many levels I have. By levels, I mean if you have, let's suppose, 10 values, or let's do a So I have here a dummy example. So I have only two types of values, but one, two, three, four, five entries. But if I convert this to a factor class, there would be only two levels, which basically means that what are the unique value types. So if I say length, of course, that would hmm, length as 5. But if I say as dot factor and that's telling me there are only two levels when you see this particular data in the form of a factor. There are only two levels, A and B. So here when I'm saying length of the levels for plate ID, which means what are the unique plate IDs that are present in my data, and there are 21 different plate IDs present across the 500 odd samples. So now I want 21 colors. And here this viridis function comes very handy. I can just say this, put this function and at the end put 21. So it would assign 21 different colors, which would be available for me to plot. Now, so what I'm going to do is, again, there is a command for you to save the values. So plot in PDF. I'm going to rather plot it here. So we can see, so I have plotted. So there is one, so in the scatter plot 3D function, there is one important parameter, that is the angle, which is at which angle this 3D scatter plot is rotated. And I would recommend definitely to rotate it to different angles to see how the gathering of the points looks from a different angle. So they get the idea, and it kind of seems that that the points which are towards the purple or the blue range, purple is kind of further apart from the the values which are on the green or yellowish range plate IDs. So this is the situation in with the raw data when we have not done any kind of normalization. So now let's see what is the status in with normalized data. So So in this case, when we are doing the PCA for the normalized data, we are not doing log2 because the normalized data is already on a log2 scale. So we do not need to do log again. That's the situation after the normalization. It seems that the points have kind of come together, are more not that separated by plate ID colors. If you plot if you save the PDF command as a whole, you would get the legend as well for the plate ID and what each of the color is for what plate ID. Okay, now what we are going to do is we are going to use the SVA package. So SVA is surrogate variable analysis. It's a well-known package for removing batch effect. It has the combat method, which is which has been referenced by many in literature for removing batch effects. That method, combat method, is implemented in the SVA package, and we are going to use that. Oh, right. So SVA didn't load here because SVA is present in a different R environment. Um, uh, so what I'm going to do is I'm going to close this session and open a new session where I have the SVA package installed. And so this is another thing. If you are another consideration, if you are doing a lot of analysis, is you would want to maintain um, for any analysis that you have kind of more or less finalized or more or less in a mature shape. You would want to freeze the version of the packages or the that you have used so that it would be much easier to write it up for a manuscript probably. And when you want to do that, you would not want to install any further packages where you in your workspace. And that brings us to the situation where you might have installation or analysis environment where you were working with 5 packages and you did some analysis, and maybe a couple of months later the line you want a 6th package, but if you want to install that package, it kind of want to install couple of other things which might ruin or break your previous installation. So this is where, as I said in the beginning, Conda environment comes plays a role. So Conda environment, what it allows you is that, as the name says, it can provide you multiple environments. So you can have within your own within your laptop, you can have multiple versions of Python, you can have multiple versions of Perl, multiple versions of R or any other analysis language, and all of them would be hosted under different environments, and they can be run they can remain on your on your system independently. So so that is that comes quite handy if you are when you started to analyze um, or you started to look at large number of data sets. So I'm going to quit this section. So here, this is my Conda graphical interface, and here you can see that I have these environments. So I have another R environment where I have the SVA package installed, and you can have as many environments that you want, and you can have a particular version of package present in that environment and something other. So that allows a lot of flexibility, um, even even if you're working on a. Taking quite long. I'll quit it and open again. Right. Um, okay, we have the SVA package is present in this environment. I'm going to go ahead with the SVA package. But first I need to load the data sets. Um, okay, so once we have loaded the SVA package, okay, to load the other stuff as well. So SVA package has the main command within the SVA package to correct for batch is the combat function that I have said, and it needs two main information. One is the biological effect that you want to retain, and then and the batch effect variable that you would want to be corrected for. So the biological variable or the biological effect that you would want to retain it goes under the um, under the object name called as mod combat. Of course, the object name as such is not fixed. You could give it any other name which you find suitable. So here what I have done is that I have given the sample class. So what is sample class in my sample info? So sample class is my information on whether these are tumor or whether these are normal. So here I'm giving the complete data for because I would want to remove the batch effect and simultaneously I would want to retain differences between the tumor and the normal. So I have said batch equal to my sample info dollar plate ID. As I said, dollar is also is a way of extracting particular columns or lists within objects. So batch is plate ID and mod. Mod is the biological effect that I would want to retain. And mean only, there is this parameter which is mean only equal to true. I'd recommend to read the help page for combat. The mean only equal to true is a parameter which is there to ensure that the batch effect removal is not too harsh, so that true biology the loss of true biological effect is minimal. That's the main parameters in the batch effect variable. Once you run it takes a couple of seconds and should be done. That's it. So now we have applied the SVA method for batch removal as well. Now we can check what is the effect, same effect of plate, the effect of plate ID after we have removed batch effect using SVA. So the same commands, but now with the batch corrected data. So that's now the same plot, similar, the same plot, but the underlying data is now batch effect corrected using SVA. I would again recommend. Um, I would again recommend that when you are looking at any data using this scatter plot to change the angles and look at the data at different angles to see if whether the points really look finally, what we are doing is we are saving the batch corrected data use .r data. This is the same file that is available on the web page as .r data file here. So that brings to the end of this particular stage. So next, what we would be doing is we would be looking at differential expression. Um, um, so hardly half an hour left for the class to go. So it basically run through the steps of differential expression. And before we wrap up, what I would try to do is go through a couple of websites and resources for you to get started in analyzing transcriptome data sets. So I'm going to quit this session now, and I would open a new session. There's roughly 20 minutes left, right? For this stage, we are going to load the stage 2 final results so that we can proceed with the differential expression. I've loaded, we've used the load function and have and here I have the batch effect collected data and the sample information, which are enough for me to proceed with the differential expression. The package that I'm going to use for differential expression is Lima, because I used boom method for normalization of RNA-seq. Um, there are other methods also available by conductor packages to normalize RNA sequencing data, like DESeq2, which is also very popular and used by many. Um, you could use either of the method, DESeq2 or Boom, or there are other packages available as well for count data normalization. Um, So for no, so what I'm going to do, so we're going to do differential expression and comparing tumor versus the normal. When I want to do that, what I would need similarly a model matrix command, which is basically a formula to tell R what are the what are the variables that I want to compare and which samples belong to which variable. So if I'm comparing tumor versus the control, so which of the samples belong to tumor, which of the samples belong to control. So that information needs to be provided to R or Lima, and that is done through this model matrix command. So here I'm saying the model matrix command, and so from my sample info, the sample class column, which has the information for which of the samples are tumor, which of the samples are control, that is taken, and so in this command, so I would again recommend everyone to look at the vignette or the manual for the Lima package. It is quite descriptive, and it explains in very lucid language the logic behind applying model matrix and how to use the how to compare two groups or nested designs or situations like this. So here lmfit is the function which is fitting a linear model based upon the formula that I have given over the data. So this is a sequence of steps that need to be done within the Lima package. So a linear model is fit, and finally a moderated t-test is applied. Um, this function, it is telling me that out of the around 30,000 around genes, how many are differentially expressed at the default p-value. So it is telling me that around 20,000 genes out of 30 are differentially expressed, which basically is an overestimation, and the reason it happens is because we have our sample size is very large, and with the statistics, if you have large sample size, I mean, if your sample sizes are very low, even minor differences would get very significant p-values. So we would probably not go, or it would not be advisable to think that 20,000 genes are differentially expressed. You would need to apply some more stringency to this. So one way to do that would be rather than so we can take the top 10% of the genes as being differentially expressed. So we can extract out the total differentially expressed gene list using this command, top table, where the final number that you give is the total number of genes that were. If you give this, then basically whatever number you give, the that many number of genes in the order of the when after ordered by the p-value, you would get that data out. So if you write 100 here, so the top 100 genes would be written out. If you write the total number of genes, so you get the total data. So p-values and log fold change for all the data. So and from there, what we could do is that we could select out the top 10% rather than going with the total. So the top 10% of of these many would be around 2,150, because that was around 21,000 total. So we can look at the how it looks. You can see that the adjusted p-value is also very high, and that is, again, as I said, indicates that if you have large sample sizes, you would have very strong p-values even after multiple testing correction, and hence much more stringency would be needed to be applied. So after this stage, once we have reached this stage, and you have the gene IDs, so these are the gene IDs which are differentially expressed, and the p-values. So after this, the next step are gene set enrichment analysis, which I would have wanted to cover, but we do not have much time left. So I would upload the command for this stage 3 and the data set for this stage 3 later on the same web page, and if you have if you wanted to come back to this over the next days, as long as the workshop is running, the pages would be available, and you're welcome to look at those commands and try it out, and hopefully when there is the next session for the for our studio, I think on the second or third day, you would have lots of questions ready. um, okay, so let's go back to the so I wanted to point out a couple of help resources for when you are analyzing data sets, genomic data sets. Of course, you would have heard about biostars. Um, this is a very, very good resource to post questions and hunt for solutions to which others might have also faced, and you could. So biostars is one, and also Stack Overflow. Um, of course, Google is the best. Google it, and if you have made a query which is or the search term is if you can put the search term wisely into Google, you would get very sensible results back, and most of the time they are good enough to move you ahead. Um, um, what else did I want to? Um, So finally, one of the things that I mean, of course, with gene expression, we have just looked at whole gene level expression differences, but there are expression differences at isoform level as well. So any given gene could have multiple transcript isoforms, and that's another level of depth or resolution that can be looked into, and in that analysis domain, there is a different method of quantifying expression, and that is the expression quantification goes by the term estimation. So generally, a expectation maximization algorithm is run, which rather than simply counting the number of reads that are present within a gene, it tries to determine that if these were the number of reads present within a gene and these were the number of transcript isoforms that are known, what could have been the level of each of the isoforms. So that's a computational problem that it tries to solve, and it gives value in the form of TPM, transcripts per million, or FPKM, and those kind of values or those estimation values are quite helpful in if you wanted to look at isoform level differences. I think we are pretty much at the end of the class now.
Thank you, sir. We have received really a nice positive response from the participants. They were really enjoying the lecture. So, sir, we have some questions from the participants if you would like to answer them.
Sure.
So, the first question we have is from David. He wants to know that if the participants wants to download like both normal and tumor data sets, can they download it from TCGA? Like both normal and tumor data sets from TCGA?
Sorry, your voice broke up in between. I couldn't get the question properly. So
Yes, sir, I can repeat it, sir. The participant wants to know that if they can download both normal and tumor data sets from TCGA.
Oh, that's great. I mean, because that was one thing I wanted to cover, but I couldn't. So, not all of the tumor cohorts or the tumor cohorts in TCGA have control samples sequenced for RNA sequencing. If you look at mutation data, of course, somatic mutation data, you would definitely need a germline sample to call. But with RNA expression, not all of the tumor cohorts do have that. But having said that, let me bring briefly to again to the presentation, and I would want to hope my is my screen visible?
No, sir, not yet.
Okay, probably is trying. Okay, okay, now it's visible. I think it's visible, isn't it?
Yes,
Okay,
sir.
so
Now.
this is Google GTech Broad. If you are not aware of this resource, this is a very useful resource. So that is this is the equivalent, normal tissue equivalent of TCGA. So if TCGA is a large collaboration for tissues from tumor, GTech is a large collaboration of tissues, normal tissues, and this is a huge, huge database, multiple tissues across the whole human tissue spectrum, RNA sequencing data available freely. Of course, it is not from TCGA, but if you are looking at a particular tissue, tumor tissue, and you are wondering if I could compare it to the normal tissue, you could get the expression data from for that tissue from for normal from here, do batch effect removal, and try to compare them and try to find out interesting differences. So that's what I would wanted I'd wanted to.
Uh, Yes, sir. Thank you, sir. So next question is like from our graduate students and postgraduate students who have interest in NGS data, and they want to go deeper in this NGS section and data analysis section. So they wanted to know that can you recommend some like online courses or textbooks or some lectures from there from where they can get some more detail or get deeper into the NGS data analysis system like that.
Um, uh, well, NGS, I mean, it depends upon whether they are looking into DNA mutation data, so mutation calling, or whether it is expression calling or copy number. So I pointed out BioStars. That's a very good resource to search for. You
Um,
can search for a particular data type analysis or a particular tool that you are interested in, and there would be multiple posts in there, which should help them to get started.
Someone wanted to know specifically about whole exome sequencing online platforms to like learn something about it.
Right. Well, I mean, yeah, as I said at the beginning of the class, that we are not doing this class on a computing environment. We are doing it on our studio. So there are many. So with whole exome or with whether it is RNA sequencing, so there is a significant amount of initial work that goes on a server environment. So with whole exome sequencing, I would want to. I mean, the next question would be, are you comfortable or in a Linux environment? If you are comfortable in a Linux environment and if you have access to servers, then I would suggest that to again go back to BioStars, and there are tutorials available there to analyze whole exome sequencing data. But if you are not, if you do not have access to your Linux environment or you are not familiar with Linux environment, then Galaxy is the answer. I think so. I think in our workshop, this workshop series, I think on the last or second last day, Galaxy is being covered. So that should be quite helpful.
Okay, thank you very much, sir. So as we know that like many of our students use either Python or R programming, but many of them also have like this doubt or confusion to like learn which computer language, like which language would be essential for data analysis, like which of the either Python or R programming, like which one is better for the data analysis in biotechnology or bioinformatics in the future. So which would be more relevant for them?
I- Well, I mean, compare when you compare Python and R has a much easier learning curve, and also because with R it is like there is a console, R Studio. You enter one command, you hit enter, you immediately see the effect. With Python, of course, with Python also you have the IPython environment where you enter a command, but um, especially for people in biological data analysis, where biology is driven mostly by visual representation. So you have most of the papers and methods making impact in a manuscript with a visual representation. So in that context, R I think is has much So like the ggplot environment, ggplot2 is a very powerful, I would say very versatile and package to create publication quality graphs. Once you are familiar with it, there is no limit to the amount of creation that you could do, and there are many add-ons that are available within R. So, with respect to biological community, I think R is much better. Um, uh,
Okay, okay. Thank you, sir. So, sir, our next question is from Sonalika, and she wanted to know that how can we filter miRNA data from transcriptome data?
Filter. I mean, so whole RNA sequence. So when RNA sequencing is done, rarely microRNAs are captured, because if you wanted to capture microRNA, you specifically do small RNA sequencing or short RNA sequencing, because the standard RNA sequencing methods capture transcripts at the somewhere above of 200 to 300 base pairs, and miRNAs don't so fall much below that length threshold. So generally, you do not capture mature miRNAs nowhere. You might capture precursor miRNAs, which are 60 to 70, but they would be very, very low. So you wouldn't need to bother about that.
Okay, thank you, sir. So, sir, we have just one last question. So this is from one of our participant, Chitta Brata, and they wanted to know like how to download batch information data from TCGA.
Okay, so let me go to the presentation screen now. I'll show how to do it. So if you do TCGA, batch, yeah, if you search TCGA batch effects, so your first hit should be this TCGA batch effects viewer. It's from MD Anderson. So this is the website. You go to launch TCGA batch effects. They have recently updated it, but yeah. So now here you can select. Let's suppose if I wanted to the TCG LUAD, so that's the lung adenocarcinoma. You select that. You select gene expression, and right, and then what is it? So here at the bottom you have the corrected you have the batch effect corrected data, or the standard data, which is extracted from where I have extracted. So you can download the batch effect corrected data directly from here, or you could download uncorrected data and try to do attempt batch effect on your own. So this is from, and all of those information like what was the tissue sequencing center, what was the shipping date, all of those information. Once you so if I click here, view analysis, so it gives you a pop-up information saying that this is the corrected data. Um, and if you download, so from here you could download the whole archive. If you click it, you would download the information, the corrected data, as well as the, or you could even visualize how much is the so this shows the effect of the batch that is present. So there are multiple different plots, and if you wanted, you could download and you could use it in your own um, analysis pipeline.
So, thank you. So, these were the questions we had from the participants, and we really had a positive response for the participants. They really enjoyed your presentation, ma'am.
Excellent presentation, Amit. I'm sure the participants have had such a wonderful session, hands-on session with you. Now I'm sure they're going to apply it in their research and get back to you with a lot of more doubts.
Yeah, sure. I mean, if they're listening, I would upload the expression, so differential expression commands and the final output from there on the same web page. And I also update some of the help resources as a separate page on there. and it should be available for the next couple of days. And if they wanted to look over the workshop days at the command for the data, more than welcome to do.
Okay. Thank you so much, sir.
You're welcome. Thank
Thank you so much.
you.
Thank you, ma'am. Thank you, everybody. So, with this, we end our first day and second session of the workshop. Thank you, everyone, for joining and showing your passion to learn new things. Have a good day, or night, or afternoon, wherever you are. So, respective to wherever you are. So, we hope to see you tomorrow again at 10 a.m. IST. And thank you for today.